We're onto the WNT pathway. This one's kind of nonsense too. I asked it to do a different. I said a cartoon. I'm gonna start doing different styles because they're all starting to look like a bunch of nonsense, all the same nonsense besides Sonic and Vin Diesel. Okay, moving. You're probably all getting sick of it at this point. You're like, oh, stupid cover slide thing. He's so into it. I need to make this entertaining for myself somehow. So, all right. Oops. How about by messing up the buttons I push and then freezing it? Okay, let's get on the PowerPoint here. There we go. All right, so the last of these signaling pathways for paracrine factors that we're going to talk about is the WNT pathway. Once WNT is secreted, it binds to glypicans here. Let's get my pointer. Here we go. To glypicans here. In this extracellular matrix, and this what this does is these glypicans, when they capture them, they cause like a local increase. They concentrate them, right? So they're kind of like gathering them from the extracellular matrix. And while it's there, it can also see, this is like kind of like supposed to be shown to be like flexible. It can also bind to frizzled, and once frizzled is bound, it causes the secretion of NODM, and NODM is a hydrolase. So what NODM will do is it will cut off WNT and remove its associated lipids. But once that happens, once it removes those associated lipids, it prevents WNT from being able to bind glycans or frizzled. So it's no longer able to bind to it. So let's look at this in the case of, that's just kind of the cycling and stuff of WNT. So let's look at how it actually does the signal transduction part. So with no WNT, in this case on the left here, this beta-catenin, which is the pink here, I had to find there's a little bit of traffic on this image. It interacts with a complex of proteins that target APC, which is this green protein here, and that gets degraded, right? So when no WMT is here, beta-catenin looks around with this axon and whatever, looks for APC. Ubiquitin is a tag that targets for degradation, and so it breaks down APC in that case. And then nothing happens. There's this TCF is repressing transcription in the normal case. So with WNT, the WNT protein binds to its receptor, which is up here. So that's frizzled. But it often does also with this LRP5/6 or LRG receptors. So usually it can just bind frizzled, but it also oftentimes incorporates both of these other two receptors as well. So the downstream transcriptional effector of WNT signaling is that beta-catenin. So it is what we're targeting downstream of this. So frizzled activates disheveled, which is a fantastic name for a gene, allowing disheveled to become an inhibitor of this GSK3. So GSK3, as you can see in the non-WNT version, is part of that complex that leads to the breakdown of APC. So here, disheveled blocks GSK3, and by blocking GSK3, it also blocks axon, because they're complex together. And by blocking that, APC is just kind of chilling. It is no longer able, or no longer inhibited by this complex. Oh, here we go. By this complex. So by inhibiting GSK3, the WNT signal frees that beta-catenin to associate with its cofactors. In this case, it can go into the nucleus and associate with TCF, and that causes it to become an activator of transcription. So in this case, TCF, I'm not quite sure if TCF, I'll admit my ignorance here, if TCF is an active repressor or if it's just a transcription factor that needs beta-catenin to be activated. Regardless, transcription's not happening, so I don't know if it really matters if it's a repressor or just a cofactor. But in the case of when WNT is bound, we get free up beta-catenin by preventing the breakdown of APC, thus starting transcription of whatever genes that need to be expressed in that tissue, right? So myelin for muscles or something like that. So that's the WNT pathway. So you can see there's a lot of them are similar. The idea is the same, kind of a ligand binds to these receptors. How they function a little bit, whether they're activating or repressing, they lead to transcription changes, etc. They generally are phosphorylated. Frizzled itself isn't, but these other it's LGR and LRP5 or 6 both are, etc. So there's some common themes there. Okay, we're gonna keep that one somewhat short. So that is it for the WNT pathway. That's our 4 pathways that we've done. So we're moving on, and I'll catch you in the next one.